Welcome Life Science learners to another installment. Trust that you guys are happy and excited, looking forward to a lesson. In our lesson, we're going to be focusing on a revision of the process of cellular respiration. We know that cellular respiration is crucial to providing energy to a cell. Let's unpack this concept, let's look at some terminology, and as we look at that, we will try and unpack some processes that are involved in cellular respiration. It's important that we have an overview of the terms, we review some of the main concepts, and we look at the process of cellular respiration in detail. Let's get straight into the core concepts. In our lesson, we're going to be spending some time defining cellular respiration. We need to look at what is the importance of energy to a cell. We will look at the process of cellular respiration, and we also need to look at types of cellular respiration where we refer to the process of aerobic respiration, which needs oxygen, and anaerobic respiration, which occurs in the absence of oxygen. We also will make reference to anaerobic respiration in plants. It's important that we recognize that there are different stages of aerobic respiration, which we will discuss in detail, starting with glycolysis, the Krebs cycle, and oxidative phosphorylation. We will also need to look at the uses of anaerobic respiration in industry. And at the end, we will try and compare anaerobic respiration to aerobic respiration. So guys, the terms that are important in this is metabolism. We often get the term metabolism used in our daily lingo. What does metabolism mean? Well, it refers to all the chemical reactions that take place in the body. These include cellular respiration and in plants, processes like photosynthesis. So metabolism refers to the various different reactions. And these all require energy, and hence the relationship to the process of cellular respiration and how metabolism is connected will be discussed in a bit. Metabolism is essentially processes that are controlled by reactions which require enzymes. These processes could either be catabolic, which is a metabolic process in which a complex molecule is broken down into simple, smaller molecules and energy being released. Metabolism also may refer to an anabolic reaction, which is the opposite of catabolic, which is a reaction that requires the synthesis of more complex substances from simple molecules. So essentially, catabolic, if you remember the word cut, so it's breaking down. And anabolic comes from building up, so ascending, climbing up. So we take simple molecules and we put them together and form complex molecules. So metabolism refers to both these reactions collectively, where we find the building up of substances as well as the breaking down of substances. Right? Aerobic respiration, guys, is respiration that occurs in the presence of oxygen. We will mention the structure called mitochondria, and that is the organelle and the site for cellular respiration. The important energy carrier molecule called ATP is the general energy carrier molecule in the cells. It's important that we define cellular respiration. Cellular respiration is the chemical process where glucose is broken down gradually in the presence of oxygen or the absence of oxygen, and that produces energy. So to recap that, so we can learn a definition very simply by looking at what is required in that process. I often remind the learners to look at a definition by drawing out an equation, in this case, the equation of cellular respiration. Let's try and do that by looking at this equation. So as I mentioned, this process produces energy in the form of ATP. We need glucose and we need oxygen, and that will produce energy in the form of ATP, with carbon dioxide and water being the waste products. So simply, it's the process that requires oxygen and glucose, and it produces the waste products carbon dioxide and water, with energy being produced in the form of ATP. And these are both the requirements and the products of cellular respiration. This can be indicated in a word equation or a chemical equation where we see a complex glucose molecule in the presence of oxygen using enzymes being broken down into water, carbon dioxide, and 38 molecules of energy-rich ATP. And so this is the word equation as well as the chemical equation that summarizes this process. It's important that you remember this equation and use this equation to understand what are the requirements or the reactants and what are the products of this process. So guys, why is energy important? Energy is important for various processes in an organism. It's required for growth, for cellular division, for the movement of organisms. It's also required for the transportation of substances, where energy moves substances into and out of a cell. And we refer to that as active transportation. So we see that energy is vital to many organisms functioning. Before we get into understanding the process, we should understand the structure of the organelle in which all of this happens. And the organelle in which respiration takes place is called the mitochondrion. This is a double membrane structure. As you would see, it contains an inner membrane and an outer membrane. And these two membranes enclose a series of folds. And these folds are called the cristae. Embedded on these folds are little structures called ribosomes. And these are the organelles that are responsible for the production of proteins. The liquid part of the mitochondria is called the matrix. And it is in these structures that various different reactions take place. Before we get into unpacking aerobic respiration, we need to know where this takes place. So aerobic respiration takes place in the presence of oxygen, inside the cytoplasm and the mitochondria of cells. Well guys, that's a wrap for this segment. We've looked at the important terms, we've looked at the overview of cellular respiration. Let's have a little break and let's get back and we will look at the reactions and how they take place. See you in a bit. Welcome back guys. Trust that you energized. Remember we're working on cellular respiration. In this segment, we're going to focus on the reactions that take place. Let's unpack those reactions by looking at a flow diagram of all these complex reactions. So guys, it's important that we remember that the stages of anaerobic respiration take place in three separate stages. The first step is called glycolysis. I often try and emphasize this by saying glycolysis, and that happens to remind me of what it does. So lysis is splitting up, and glyco refers to the glucose. So try and remember it that way. So this takes place in the cytoplasm. The second stage is called the Krebs cycle, and this takes place inside the mitochondria. So in the cell, we saw that cytoplasm is the one process where it happens called glycolysis, and now we see that inside the mitochondria we have the Krebs cycle. And the third stage is called oxidative phosphorylation, and we'll unpack that in a bit. And this takes place also inside the mitochondria. So there are three stages. The first stage happens in the cytoplasm, which is called glycolysis. The next two, which are the Krebs cycle and oxidative phosphorylation, take place within the mitochondria. Right, so as we look at this diagram, I'm going to very quickly circle out the different stages. As you see here, glycolysis is indicated with the number 1, and here we have an outer view of the mitochondria, which shows you the second stage called the Krebs cycle, which then in turn supplies products to the third stage called oxidative phosphorylation. And as I mentioned, both of these occur within the mitochondria. So guys, we will look at what is produced in a bit. Let's get straight into looking at this reaction called glycolysis, which is in the cytoplasm. So guys, as I mentioned, glycolysis refers to the splitting up of glucose. So this takes place outside the mitochondria in the cytoplasm of the cell. So as we look at the cell, we know that outside the mitochondria we have the cytoplasm. So we're in the cytoplasm here. And this is where glycolysis is taking place. As I mentioned, it's in the cytoplasm. No oxygen is required during this stage. So it's again a process that does not require the oxygen. The next step of the process is when glucose is broken down. As I mentioned, the word lysis refers to breakdown or splitting. So the glucose is broken or split into smaller molecules. And in that process, they release small amounts of energy that is stored in energy-rich ATP molecules. So if we look at that, we can see that energy that is released from the breakdown is then used to produce energy-rich molecules called ATP. At the end of glycolysis, we see the production of pyruvic acid. So the glucose molecules split up into two molecules of pyruvic acid. The pyruvic acid then moves into the next stage, which is the Krebs cycle, into the mitochondria, and the energy-rich hydrogen atoms, which are now in the form of ATP, are used in the next steps. Right, so that's glycolysis. We mentioned that the energy-rich hydrogen atoms are then used in the third stage of the process. Let's look at this in a greater detail. So we're seeing that the energy-rich hydrogen atoms then move into the third step, and the two pyruvic acid molecules now move into the first step of the Krebs cycle. We must note that at this stage, only two molecules of ATP are produced. The second stage is called the Krebs cycle. This can only take place if oxygen is present. So we know that oxygen is required for respiration. That oxygen is required in the second stage, which we refer to as the Krebs cycle. So now we see the importance of oxygen coming into play. This occurs inside the mitochondria. This process also releases carbon dioxide and high energy hydrogen atoms. So we look at the energy-rich hydrogen atoms being released. We're seeing that it requires oxygen, and we also know that it produces carbon dioxide as a waste product. So let's read further. Transports hydrogen atoms to the third stage, which we see here, and that goes to the third stage of oxidative phosphorylation. And that happens through an energy carrier enzymes. So we got this hydrogen-rich atoms are carried through by a complex molecule into the next stage. Right, and so we're seeing this further again, moving into the oxidative phosphorylation reaction. Let's look at the third step, which is oxidative phosphorylation. So guys, this takes place again inside the mitochondria and requires oxygen. We see that oxygen is clearly needed for this process. This passes the high energy hydrogen atoms from one hydrogen energy carrier enzyme to the next, and as it does that, it releases energy slowly. So if you look at this, it gradually passes the energy from this in a series of events, releasing water and producing ATP in that reaction. So we find that it uses the released energy, it combines the energy where it takes an adenosine diphosphate and adds a phosphate. And hence we refer to this process as phosphorylation, essentially adding a phosphate to a diphosphate to give you a triphosphate, which is called ATP. And that reaction can be simply illustrated where ADP plus a phosphate gives you this energy-rich molecule called ATP. Okay. The oxygen that is used in this reaction acts as the final hydrogen acceptor. So the oxygen that comes in will now combine with the de-energized hydrogens to form what we refer to as water. So the hydrogen atoms that have given off the energy now combine with oxygen and they form water, which is then released as a waste product during cellular respiration. So again, the importance of oxygen is to combine with the de-energized hydrogen atoms to form water, which is a waste product of cellular respiration. The next concept that we're looking at is anaerobic respiration. If we just step back, we said that this was aerobic respiration where oxygen was needed. It's important that we now look at the difference between aerobic and anaerobic respiration. Remember that anaerobic respiration occurs in the absence of oxygen. So again, let's see how that process is different. The key terms is that anaerobic respiration refers to the process occurring in the absence of oxygen. It's also called fermentation. In plants, this type of anaerobic respiration is occurring in yeast and other cells, and we often refer to it as alcoholic fermentation. Alcoholic fermentation is the breaking down of glucose in the absence of oxygen to give rise to the production of alcohol in plant cells. So we know that this is commercially used in industries in the production of alcohol. So we look at that in a bit. When anaerobic respiration occurs in animal cells, we refer to that as lactic acid fermentation. This is the breaking down of glucose in the absence of oxygen to form lactic acid in animal cells. So guys, the simple analogy for being able to remind you of what happens when you have cramps is the absence of oxygen during cellular respiration. And the buildup of lactic acid is due to insufficient oxygen during cellular respiration. So the body then resorts to carrying out respiration in the absence of oxygen to produce energy. And hence, while producing some energy, it also produces what we refer to as lactic acid. And that is the chemical compound that builds up in muscles, causing the cramping in the different areas. Right. So let's look at anaerobic respiration and where it takes place. Again, as I mentioned, this takes place in the absence of oxygen, so without oxygen. It generally occurs for short periods of time, mainly during physical exercise. And this is again a respiration or a reaction that ensures that some amount of energy is produced when the demands of the body's oxygen are high. This occurs differently in plants and animals to produce less ATP than anaerobic respiration. Aerobic respiration produces 38 molecules of ATP. However, anaerobic respiration produces only 2 molecules of ATP. And so we see a significant difference. And again, this is a mechanism that ensures that there is some energy produced in the absence of oxygen. Okay, the lactic acid can be converted back into pyruvic acid when the muscles receive oxygen. And so often, if you're exercising after a strenuous workout or a game, we often refer to coaches saying, well, warm down. And warming down essentially means let's continue to inhale and bring in more oxygen into our muscles. And that oxygen then helps to revert back into cellular respiration using oxygen, and therefore converting that lactic acid back into pyruvic acid, which then can be used to produce more energy that is needed in the presence of oxygen. So it's important that when you do exercise that you understand why we experience the stiffening of our muscles, and that when you warm down, it's increasing the oxygen back into your muscles after a strenuous exercise, so that we can prevent the buildup of lactic acid in those muscles. We also know that anaerobic respiration in animal cells, as I mentioned, called lactic acid fermentation, occurs in muscles during intensive exercise. And this enables what we call glycolysis to take place in the cytoplasm in animal cells. As I mentioned, those crystals build up and cause the stiffening and cramp of those muscles as they build up in the cytoplasm of the cell. This results in the accumulation of lactic acid, causing muscle cramps and a lot of discomfort and pain. During this process, only a small amount of ATP is produced. However, this process in plants occurs slightly different. As I mentioned, it's called alcoholic fermentation. And this enables the glycolysis to occur in the cytoplasm of plant cells. And this in turn builds up what we refer to as lactic acid. Let's see what happens to that lactic acid. Let me go back. Pyruvic acid, my apologies. This further breaks down the pyruvic acid, forming ethanol, which is then releasing carbon dioxide in that process. This reaction can simply be summarized where we look at glucose being broken down in the absence of oxygen to produce ATP and pyruvic acid. This further is broken down into ethanol and carbon dioxide. And we see this reaction being the release of ethanol, being called alcoholic fermentation. Yeast and other fungi respire anaerobically, and these are used to produce alcohol as beverages in the beer and wine industry. This process is of very importance and significance in the industry where we refer to the brewing of alcohol. Yeast cells are also used in the baking industry where we see that bread is able to rise during the baking process. This is called fermentation, and it's basically able to use in the production of cheese as well. The use of anaerobic respiration in yeast and fungi produces alcohol as well as beer and wine. Yeast cells can be used in the industry during the baking of bread. We also know that we have cheese that is produced as a byproduct of fermentation. So as we wrap this section up, guys, we're looking at the use of anaerobic respiration, which can be used to produce soy sauce, and that is a product which is produced during anaerobic respiration from lactic acid buildup. We also know that lactobacillus can produce cheese during anaerobic respiration, and then Saccharomyces, which would be yeast, is used in the production of ethanol, wine, as well as the baking of bread. Welcome back, life science learners. In our segment today, we're focusing on the revision of cellular respiration. Let's apply our understanding to various different questions. I remind you to make note of some of these important concepts. Write them down so that you can apply them when you're working through some questions. Let's get straight into the lesson and apply our understanding to various different questions. Question 1. As I always do, start off with some multiple choice questions. In this segment, it's important that we try and understand some basic concepts as we unpack them using cellular respiration. So the first question, cellular respiration in a green leaf takes place A, during the day only, continuously B, C, during the night only, and D, during photosynthesis only. Guys, as I mentioned earlier on, cellular respiration is a fundamentally important process for the production of energy. All living organisms require energy throughout their life. And we find that cellular respiration is a process that occurs continuously. And that means during the day and during the night. So this will take place continuously as we require energy consistently being produced. Let's look at the next question. The following components are involved in cellular respiration, and we know that energy is the product, carbohydrates are needed as the reactant, we know that carbon dioxide is a waste product, water is needed, as well as oxygen. So we're looking at various different components involved in this process. Let's read the question further. Which one of the following combinations show the correct way in which these components are involved? So guys, this question gives you various different components. It's important that we go back to these components and look at what is needed. So we talk about the reactants and the products. And so guys, we know that one of the products of photosynthesis respiration is energy. So that's going to be a product. Carbohydrates are the reactants. So we know that those are the reactants. Carbon dioxide is a product. We know that oxygen is required, so that's a reactant. And we see water as a product. So we've got, we've got products, which are energy and water, and we've got reactants, which are carbohydrates and your oxygen. So when we unpack this question, it's important that you look at that. So it's going to be those two reactants forming those three products. And as I mentioned, if we go back, it's 2 and 5. So 2 and 5, guys, let me just go back one more slide. So it's 2 and 5, which were, as I mentioned, your reactants. So it's your carbohydrates and your oxygen, which would be on this side of the equation, with the rest coming onto the left-hand side. So this equation required that you understand what the reactants were and the products. So a very different way of testing a very simple concept by mixing up the combinations. So it requires that you read. Let's move on to 1.3. The conversion of pyruvic acid into lactic acid occurs during. So guys, we've looked at the process of cellular respiration. We know that during anaerobic respiration, we find that in animal cells, the pyruvic acid can be converted into lactic acid, producing some energy. Let's look at the options. So does it occur during photolysis, or glycolysis, or anaerobic respiration, or oxidation of glucose? So guys, it's important that we know that this reaction happens during glycolysis. So when glucose is broken up, it's broken up into 2 pyruvic acid molecules. That is the process that occurs during glycolysis, and that gives you, in anaerobic respiration, lactic acid. So the answer there was B. A phase that does not require oxygen during cellular respiration. Guys, there are various different stages that we've discussed. So again, the focus is does not require oxygen. So guys, we've looked at the Krebs cycle. We've looked at glycolysis as a reaction in which glucose is split up. So what would be the answer? The answer is glycolysis, because that is the reaction that takes place outside in the cytoplasm, independent of oxygen, where at least 2 molecules of pyruvic acid are produced and ATP is produced. And we said that these were reactions that were ensuring that even in the absence of oxygen, that some energy was produced. Let's move on to question 2. This relates to biological terms. So we'll go through a few of these and apply our understanding. The process during which glucose is converted into pyruvic acid. I'm going to give you a moment to write your answers down. Let's go through them, and as we do that, I hope that you'll be able to give me 3 answers in a bit. The second question is, the reagent used to test for the presence of carbon dioxide. So guys, when doing respiration experiments, we often have carbon dioxide as a waste product. What is the chemical reagent used to test for the presence of carbon dioxide? I'll give you a moment to think about that. Question 2.3. The acid that accumulates in the muscles of humans during continuous, strenuous physical activity. And we refer to that as anaerobic respiration in animal cells, where in the absence of oxygen, respiration does occur, but it builds up in muscles causing stiffness. So I hope that rings a bell. Now let's reveal those answers. So as I mentioned, the first answer would be glycolysis, and that is when the glucose is converted into pyruvic acid. The second question was relating to what chemical is used to convert or to test for the presence of carbon dioxide, and that is clear lime water. And we know that clear lime water, when carbon dioxide is introduced into that, turns milky, and so that could be an indicator for the presence of carbon dioxide. And finally, we looked at lactic acid being built up in the muscles during anaerobic respiration, and that is what causes the stiffness in muscles. We've got 3 more and we'll wrap this segment up. The gas which is essential for the Krebs cycle to occur. We know that the Krebs cycle is the second stage that we look at. I'm going to give you a moment to reflect on that. 2.5. The folded structures found on the inner membrane of the mitochondria. As a quick recap, we know that there are folds on the inner membrane, so this points to those folds. And 2.6, the stage of anaerobic respiration that releases carbon dioxide. So we looked at those a little while ago. Let's see what your answers were. So as I mentioned, the gas which is essential for the Krebs cycle is oxygen. And we know that oxygen is a requirement, and that is the only reaction that requires it. It's the first reaction of the Krebs cycle. The folded structures on the inner membrane of the mitochondria are called the cristae. And finally, the stage of anaerobic respiration that releases carbon dioxide is the Krebs cycle. So guys, we've spent some time unpacking some of the terms by using some questions that required us to analyze various options around the different reactions. We've also looked at some descriptions that test the understanding of the process. And these again are important in being able to understand and unpack the processes that we've just looked at. Let's have a little break, and when we get back, we will take this to the next level by applying this to more complex questions. See you in a bit. Let's re-energize and start again. Welcome back, life science learners. In our segment, we're going to wrap up our understanding of cellular respiration. We're revising cellular respiration, and the best way to do that is by applying our understanding to solving a few questions. I encourage you to enjoy this lesson by interacting. As I go through these questions, I'll give you an opportunity to think about these answers, and if you can jot them down when we go through these, I hope that you can reflect on your answers and see if you were spot on. So let's attempt a few more questions on further application of cellular respiration. Question 4. A baker is testing a new recipe in order to bake the perfect loaf of bread. The 2 main ingredients in the bread dough are flour and yeast. She divides the dough into 3 equal parts and subjects all the loaves to different treatments before baking. Loaf A, she immediately bakes the bread. Loaf B, she covers the dough with a damp cloth and leaves it in a warm area for an hour before baking. Loaf C, she covers the dough with a damp cloth and leaves it in the fridge for an hour before baking. After baking the 3 loaves, she measured the height which each of the loaves had rose. The results are shown below. Loaf A, as indicated here, doesn't look like it has increased significantly in height. Loaf B has a bread that has risen very handsomely, and loaf C seems to be a moderate amount of height increase. So essentially this experiment is looking at the effect of different factors. And if you go back to the question, the different treatments are seen where the individual has kept them in a warm environment, then in a fridge, and then she's immediately used or placed them into the oven. So what's important in that, we've identified that there is the effect of different treatments on the baking of the bread. So she's mixed the dough with yeast and flour, and she's allowed that to stand for a little while in different temperatures. So let's formulate a hypothesis for this investigation. This is a 2-mark question. Guys, when putting together a hypothesis, it's important that we first link our hypothesis to the aim of the experiment. Remember that a hypothesis is often an educated guess that indicates what the possible results might be or anticipated results may be. It's important that when you string together the hypothesis that you include the independent variable, that is what is being tested, along with the dependent variable in terms of what is being measured to draw a conclusion. In this particular experiment, we know that the different treatments in terms of temperature is what is being tested. And the way in which we measure the effectiveness is by looking at the height, as she mentioned here, measure the height. So the height is being measured as an indication of how much the bread has risen. And that often is an indication of whether that method or treatment was effective. So, a possible hypothesis would be bread dough kept at warm areas will rise the highest after baking. Or you can also say that the bread dough kept in the coldest area or the cooler area will rise the least. And so what we've identified is that we've identified the independent variable, which is the concept of temperature, as well as the dependent variable, which is the measurement of the height in terms of how much the bread rises. So that is a hypothesis. Again, it is a statement and not a question of intent, where you say to investigate or to determine or to find out. All of those would be the aim of the experiment. Let's move on to question 4.2. The question requires us to identify A, the dependent variable. So I want you guys to think about that. So the dependent variable is that variable that is used to be able to draw a conclusion. So what was measured in this experiment that helped us to draw a conclusion about the effectiveness of different temperatures? I'll leave that to you to think about. The next part was identify the independent variable. And I often remind my learners to think about this question when trying to establish the independent variable. What is being tested in this experiment? We're testing the effect of some factor on the process of the bread dough rising. So think about that. And finally, one controlled variable. A controlled variable in this experiment would be something that is subjected, that you keep constant and subject all the different testing samples to. So a control variable is used to ensure that we have a fair test. So there was one variable that was kept constant in this experiment. So let's unpack those answers now. So the dependent variable in this case is the height of the bread. So in order for us to determine which was the most effective method of treatment, we measured the height of the bread. And so that was helping us to draw a conclusion. And we were able to say, well, that's probably the most effective method that was used to get the highest, or the loaf of bread to rise the highest. So the height of the bread is the dependent variable. The independent variable is the different temperatures. As we mentioned, the different treatments. In this case, there was immediately placed into the oven. In loaf B was placed in a warm environment for an hour, and loaf C was placed in a fridge. So it's the different treatments, and in this case, it is the different temperatures if you think about it as a variable. And finally, the control variable was the same amount of dough has been used. So it also mentions that the dough was divided into 3 equal parts. And this ensures that all the amount of dough was equal. So when we measure the height, we know that the height is in relation to the initial amount being the same. Which loaf do you consider to be the control for this experiment? Give a reason for your answer, and that's a 2-mark question. Guys, it's important that at this point we reflect on the difference between the control and a control variable. In any experiment, we have variables that we keep controlled, which all the test samples are subjected to, and that we refer to as a control variable. As you've just seen, we've spoke about the dough being divided equally, so the amount of dough was equal, and that's a control variable. However, this question requires us to analyze what was the control for this experiment. And guys, the best way to identify the control in any experiment is to ask yourself the following question. Which of the different samples were not subjected to the independent variable? Meaning, which of those different test tubes or samples, in this case loaves of bread, were not subjected to the different treatments? We know that the loaf of bread B was kept in a warm environment. Loaf of bread C was kept in a cooler environment in the fridge. Loaf A was placed immediately into the oven. So that was subjected to no specific treatment. So the rationale for saying that A was the control was that it was not exposed to any temperature, but baked immediately, meaning that it was not subjected to the independent variable of different treatment. And so it's important that you differentiate what a control is and what a controlled variable is. And so having a control in any experiment ensures that your test is reliable and it produces results that are accurate. Let's move on to question 4.4. One of the main ingredients is yeast. Name the function of yeast in this investigation, and that's a 1-mark question. So guys, the main ingredient having yeast in the experiment is to ensure that the process of cellular respiration takes place. During cellular respiration, we know that carbon dioxide is produced. When we bake bread, the carbon dioxide is released, and that is what causes the dough to swell up. And that is what makes the bread fluffy and moist. So when you cut through a loaf of bread, you often see pockets or little holes in it. And that is the air bubbles that are produced during the fermentation or during the yeast producing carbon dioxide. And those bubbles accumulate and cause the dough to swell up. And that's essentially the significance of having the carbon dioxide being produced by yeast. Which biochemical process allows the yeast cells to perform its required function? And so guys, we've been looking at cellular respiration. So it's a process of cellular respiration that is causing these bubbles to be produced. And finally, what is the yeast source of glucose? So the yeast cells require a source of glucose to be able to carry out cellular respiration. If we look at it, there is flour that is provided, and flour is a type of carbohydrate which is a complex form of glucose. So the glucose is going to be provided by the flour, which again contains carbohydrates. So the answers to these questions would be, again, the starch is present in the flour. We know that that process that we've just discussed was cellular respiration, and the process of using sugar is that the carbon dioxide causes the bread to rise. Right. What conclusion can be made from the results of the baked loaves? So guys, when we've looked at this, we measured the height of these loaves, and we can clearly see that the height of bread loaf B was significantly higher than loaves C and loaf A. So what is the conclusion? The conclusion needs to relate to the aim of the experiment. The aim of the experiment was to determine the effect of different temperatures. So let's link our aim to that. So it's got to link to the effect of the temperature or the different treatment. So this loaf B was subjected to 1 hour of being placed in a warm environment. And so when bread dough is left to rest in a warm environment, it will cause or result in bread rising significantly higher as compared to bread that is kept in a cooler environment. So I'll string that together in my answer here. So as I mentioned, the bread dough that must be kept in a warm area for a period of time before baking so that the bread can rise. Okay, and finally, how can the bread maker improve the reliability of her experiment? Guys, it's important that we're able to reflect on any experiment and comment on the accuracy of our results as well as the reliability. In this particular experiment, the baker had separated the dough into 3 different parts. All she did was use 1 dough for each of the different temperatures. In order to improve the reliability of her results, it would mean that she could repeat the experiment and see if she got the same results, or she could increase the amount of dough and have 2 lots of dough for sample A, sample B, and sample C. And that would mean that you would be able to see if those results are consistent. So guys, we've looked at the application of cellular respiration, we've unpacked the different stages, and we've looked at how it's used in industry. I trust that you've enjoyed the lesson, and I hope that you can go back, reflect on what we've just done. From my side, cheers, have a bio-lekker day.